Then 26th question. Which of the following event requires ATP? See the graphics of that process and see. ATP, number 1 during regeneration, number 2 during reduction, number 2 during reduction. So answer is very clear, that is during the reduction and regeneration. Reduction and regeneration. These two processes are required. These two processes require ATP. During reduction, they are required by 3-PGA. 3-phosphoglyceric acid uses ATP of light phase, and get converted into 1,3-dipGA. 1,3-dipGA. This is the first compound which uses the ATP of light phase, and during the regeneration, ribulose 5-phosphate is formed. Ribulose 5-phosphate is formed. This ribulose 5-phosphate requires ATP for the formation of what? Ribulose 1,5-biphosphate. 1,5-biphosphate. This is the second compound which needs ATP of light phase. Second part is from regeneration and the first part from reduction. The number of ATP and NADPH2 consumed during the fixation of 1 CO2. Actually, for the 6 carbon dioxide, for 6 CO2 in C3 plants, 18 ATPs are required and 12 NADPH2 are required. Now the position is very clear. That is for 1 CO2, 3 ATP and 2 NADPH2. So the answer for this question is 2, that is 3 ATP and 2 NADPH2. Sugarcane, direct answer C4 plant. One of the very good example of C4 plant from which C4 cycle was studied. Sugarcane plant, Saccharum officinarum. Sugarcane, Saccharum officinarum. Scientific name of sugarcane plant, Saccharum officinarum. Sugarcane. And this plant is known for the discovery of C4 cycle. Other C4 plants are Z maize, that is normal maize plant. Second, sorghum. Second example, sorghum, essential for examination. And some grasses. And amaranthus. These are all the examples of C4 plants. Primary acceptor of carbon dioxide. There are 2 CO2 acceptor in C4 plant. In C4 plant, there are 2 CO2 acceptor. 2 CO2 acceptors are there. In mesophyll cell of leaf, PEP is there, phosphoenol pyruvate. PEP, 3C compound, it is there. Then second is RUBP in bundle sheath. RUBP in bundle sheath chloroplast. This is bundle sheath chloroplast. So double CO2 acceptor are there, but the question is asked, that is primary acceptor of carbon dioxide. In that case, PEP is correct answer. PEP traps carbon dioxide and form oxaloacetic acid 4C compound as first stable product. So initial acceptor of carbon dioxide is PEP. Otherwise, simple acceptor is asked answer 2, that is PEP and RUBP both. PEP 3 carbon compound and more sensitive than RUBP. PEP is 3 carbon compound and more sensitive than RUBP. C4 plant sugar formation. Actually, C4 cycle takes place in two structure. C4 cycle takes place in two structure. Part 1 is mesophyll cell, then part 2 is in bundle sheath cell. Part 2 is in bundle sheath. In mesophyll cell, H-Slack cycle is in operation. H and Slack cycle. H and Slack cycle takes place in mesophyll cell, whereas in bundle sheath Calvin cycle takes place. And sugar formation is done by the Calvin cycle. Sugar is formed by the Calvin cycle. So in C4 plant, synthesis of sugar or final CO2 fixation takes place. Answer is bundle sheath cell. Bundle sheath cell. 31. H and Slack cycle is found in direct answer, that is C4 plant. Direct is C4 plant. MDH. Second fellow CR Slack. Both are Australian. They discovered another pathway of CO2 reduction. Another pathway of CO2 reduction in some plants. These plants are known as C4 plant, and the pathway is now named as H and Slack cycle. H and Slack cycle operates in C4 plant as initial cycle of CO2 reduction. It takes place as initial cycle. H and cycle operates as initial cycle of carbon dioxide reduction. Carbon dioxide reduction. This is H and Slack cycle. 32nd question. First stable product. See the cycle. Phosphoenol pyruvate 3 carbon compound in mesophyll cell. Traps atmospheric carbon dioxide, that is atmospheric carbon dioxide is taken up by PEP. This reaction is known as carboxylation. And oxaloacetic acid 4C compound is formed as first stable product. So answer is oxaloacetic acid. And that's why these plants are also called C4 plant, because of the production of oxaloacetic acid directly by the carboxylation of PEP in mesophyll cell. 33rd question. Which of the following is common in C3 and C4 plant? Yes, Calvin cycle is common in both, because sugar is formed only by Calvin cycle, and sugar formation is the aim of C3 plant and the C4 plant both. And for the sugar formation, Calvin cycle is in operation. That's why it takes place in all photosynthesizing plant. Calvin cycle takes place in all photosynthesizing plants. Next one. C4 plants have advantage over C3. That is, they can perform photosynthesis in hot and dry habitat also. They are well adapted for the tropical climatic conditions. C4 plants are well adapted for the tropical climatic condition where the temperature is comparatively high. They are adapted in the tropical region, and even they are perform photosynthesis in this range also. So they are suitable for the hot and dry climates. 35. Which pair is wrong? C3 plant, maize wrong. C4. Maize is C4 plant. Maize is C4 plant. Crans anatomy is found in their leaf. What is crans anatomy? This is the section of leaf. This is the vascular bundle area. And you see the ring-like orientation of mesophyll cells around vascular bundle. This is the mesophyll cells in the leaf. They are arranged in ring around vascular bundle. This is Crans anatomy. This is also called rat anatomy. Crans anatomy or rat anatomy, that is arrangement of mesophyll cells around vascular bundle in ring. That's why this anatomy is called Crans anatomy or rat. This is also another word is there, rat anatomy. That is rat anatomy. Then Kelvin cycle, yes, is for stable product is 3 pg. HN cycle for stable product is oxaloacetic acid. This is 4C compound, this is 3C compound, and right. And the first one, that is maize, is C4 plant, not C3 plant. So this pair is wrong. Correct answer is C4. PEPCO, full name PEP carboxylase. PEP carboxylase. It is associated with the CM plant as well as C4 plant. In these two cases, this enzyme is needed because it is a primary acceptor of primary carboxylation. PEP, CO stands for carboxylase, needed for, it is required, it is needed or required for primary carboxylation, in C4 and CAM plant as well. So answer is more than one option. Primary acceptor I have already stated, that is PEP. In this plant, PEP. Phosphoenol pyruvate. 3 carbon compound is that, and at night, that is in night, these plants actually synthesize organic acid at night. In night, these plants undergo the reduction of carbon dioxide. They perform reduction of carbon dioxide at night, and this PEP traps atmospheric carbon dioxide, first of all converted into oxaloacetic acid. This oxaloacetic acid reduced undergoes reduction to form malic acid. So C4 plants, they form malic acid at night. They form malic acid night because of this reaction. And this reaction involves the reduction of carbon dioxide at night. So primary acceptor, because during day, in these plants CO2 reduction takes place internally when carbon dioxide is released from the malic acid. And that carbon dioxide is not from the atmosphere. Atmospheric CO2 is trapped at night when stomata are open. So one more speciality of CAM plant, that is they have scotoactive stomata. Scotoactive active stomata are found in these plants. That is, they open at night. When they open, then only CO2 reduction takes place. Photoactive stomata, that is, they open at night. And this is the feature of CAM plant. And when they open, then only reduction of carbon dioxide is possible. 38. Photorespiration. In normal process or normal course, in C3 plant, RUBP if undergoes Kelvin's carboxylation, then only photosynthesis is possible. But this RUBP has dual nature. This RUBP has dual nature, that is sensitive to carbon dioxide and oxygen as well oxygen. Sensitive to carbon dioxide as well as oxygen. So if CO2 is not available in sufficient manner, and atmosphere is already rich in oxygen, if CO2 is not available up to the mark, in that case oxygen is already rich in the atmosphere, this RUBP undergoes conditional oxidation. The RUBP undergoes conditional oxidation during the daytime. So instead of reduction of carbon dioxide, it is evolved in this process. This activity is called photorespiration. That is conditional oxidation of RUBP. Conditional oxidation of RUBP in presence of light. When there is a low CO2, CO2 is not available up to the requirement, and oxygen is already high in the atmosphere and high O2. This is the condition for photorespiration. The process accomplishes in 3 cell organelle, and in order to their participation, this is the question, and the answer is chloroplast, peroxisome and mitochondria. Photorespiration takes place or accomplishes in these cell organelle. It starts with the chloroplast. Chief metabolite. See here. RUBP. When it undergoes oxidation, it forms 2 products. One is phosphoglycolate. And 2 is 3-PGA. But this phosphoglycolate immediately loses phosphate and converted into glycolate. This activity takes place in chloroplast. It is the first 2 reactions of photorespiration. And this glycolate is C2 compound. Its formation as well as consumption both are seen in photorespiration process. That is formation of glycolate as well as consumption. Formation of glycolate as well as its consumption. It is seen in photorespiration. That's why photorespiration is called glycolate metabolism. It is called glycolate metabolism or C2 cycle because it's a C2 compound. So final answer for this question, chief metabolite of this process is glycolic acid or glycolate. Another term for the photorespiration I have just described, that is glycolate metabolism or C2 cycle. And for the first question, answer again at the time of description of photorespiration I have given. that is high O2, low CO2. Low CO2 means below compensation point. Low CO2, that is below compensation point. What is low CO2? That is below compensation point. And what is compensation point with regards to CO2? That is rate of photosynthesis is equal to the rate of respiration. Rate of photosynthesis, if it is equal to rate of respiration, that is compensation point. If it is with respect to the CO2, it is called CO2 photosynthesis compensation point. So there is a limit of carbon dioxide, that is C3 plants can perform photosynthesis when carbon dioxide is available at least 50 ppm in the atmosphere or surrounding the plant. Part per million, 50 per million, that is CO2 compensation point for the C3 plant. That is CO2 compensation point for C3 plant is 50 ppm, that is high, and it is C4 plant for 1 ppm to 10 ppm, that is quite low. That is C4 plants are able to perform photosynthesis when very low amount of carbon dioxide is there in their surrounding, but C3 plant can't. So this is the photorespiration, that is low CO2, that is below compensation point. High oxygen is already high. Oxygen acts as inhibitor in photosynthesis in C3 plant. Oxygen acts as inhibitor of photosynthesis in C3 plants. 42. During photosynthesis, oxygen in glucose. Photosynthesis requires carbon dioxide plus water. And you see, oxygen is contained in the carbon dioxide as well as water both. But in photosynthesis, we have already studied that C and O are contributed by carbon dioxide, only hydrogen is provided by the water. So hydrogen is provided water, that is water undergoes oxidation, then only hydrogen ions are released. These are trapped by carbon dioxide, then only C6H12O6 is formed. The answer is very clear, that is oxygen in glucose come from CO2, not from water. CO2 compensation point in C3 plant is high. I have already mentioned in the previous description, and that is 50 ppm for C3, 50 ppm, and C4 plant 1 to 10 ppm, a very low CO2 compensation point. Blackman's law of limiting factor applicable for photosynthesis. That is, if a process is affected by number of factors like carbon dioxide, temperature and light, suppose they have the effect over the photosynthesis process, then the rate of process is regulated by that factor which is in minimum. In those conditions, again, law of limiting factor denotes if a process of photosynthesis affected by number of factors, carbon dioxide, temperature and light, then that the process is regulated, the rate of the process is controlled by that factor which is in minimum in those conditions. That factor is called limiting factor. Only that factor is known as limiting factor. This is law of limiting factor. Then the final question, which of the following is not the limiting factor for carbon dioxide? That is oxygen. Carbon, light intensity and light quality, these two are the limiting factor. Light intensity, light quality. Light intensity, then light quality, but not light duration. These two with regards to the light. Carbon dioxide is natural limiting factor. This is already natural limiting factor in clear days. This is natural limiting factor in clear days. That's why if carbon dioxide increases in the atmosphere, that is useful for C3 plant, not for C4. C3 plants, they flourish well. They flourish well. This is carbon dioxide fertilization effect. Carbon dioxide fertilization effect. This is all about the process of photosynthesis in green plants. Thank you.